move to last and very important topic which is remaining. It is clinical trials. Clinical trials. Now currently we have thousands of drugs in the market. The question is how they came into market. Now suppose I do a research in my laboratory and I make a drug which is very good antihypertensive drug. So I make 1,000 tablets of that and go to chemist to sell this. So can the chemist sell those drugs? Obviously not. Why not? Because drug is a chemical substance. It can even kill someone. So who will be responsible? So before going into market, there must be extensive testing on the drugs for their safety and for their efficacy. So first of all, what we do is we test the drug on animals. We take mice or rabbit, we give them the drug and see whether they are effective or whether they are safe. If a drug is found to be safe and effective in animals, then can we sell the drug to the chemist? Obviously not. Because we have tested in the animals, we need to test in the humans also. And that testing of drug in humans is called clinical trials. Testing of drug in humans is called clinical trials. And clinical trials are done under 4 phases. Phase 1, 2, 3 and 4. So first of all, phase 1 trials are done. And phase 1 trial, the special thing to remember is they are done on healthy humans. Phase 1 trials are done on healthy volunteers. The question is, we have to give a drug to a patient. Then why to test the drug on healthy person? Okay, healthy person, will they not develop side effects? Obviously they can also develop. Then what is the logic is, a healthy person has a good immune system. So that means even if some side effect occur, the body can manage. Whereas a diseased person may die because of that side effect. So phase 1 trials are mainly done for in healthy people. The question is what we do. What we do in phase 1 trials. Or more importantly, what we cannot do in phase 1 trials. The important thing is, in phase 1 trials, we never do efficacy testing. We cannot do, in fact. Like, we are developing a new anti-tubercular drug and we give it to a healthy person which do not have tuberculosis. So can we know whether the drug is efficacious or not? Obviously not. So phase 1, we cannot do the efficacy. So when we cannot do the efficacy, what we will do? Safety. Obviously we do safety. But anything more important than safety? Asking the question in other format. Before phase 1 trial, how many humans have received this drug? None. That means no person has ever received this drug. No human have received this drug. So when it has not been given to humans, then how we will come to know that what dose to give? So that is the main aim of phase 1 clinical trial. Phase 1 clinical trial, we find the dose of the drug. And how to find the dose of the drug? So what we do is, from the animal studies, we know that we extrapolate that this is the human equivalent dose. So, but that dose comes into a big range. Like we can give from 50 to 500 milligram. So we'll call one healthy person, give him the lowest dose in that spectrum. In this case, 50 milligram, and we'll check all the tests on that person. Like we'll do hemogram, LFT, KFT, ultrasound and all the tests. If there is no problem, we will send him home and then we'll call the another person and give him higher dose, 100 milligram. Do all the tests, no problem, we send him home. Next person will give the still higher dose. So in this manner, we will keep on increasing the dose and we will stop at that dose where the intolerable side effects come. So the dose which is lesser than that is called as maximum tolerable dose. So as the name tells, it is the maximum dose that can be tolerated by humans. So the main aim of phase 1 clinical trials is to know the maximum tolerable dose of a drug. So phase 1 is to know the maximum tolerable dose. Now there are some conditions or some drugs where if we give to healthy person, they are also likely to develop serious side effects, like anti-cancer drugs. If we give anti-cancer drug to a healthy person, even they may develop bone marrow suppression. So it will be totally unethical to expose the healthy person to such type of drugs. So in those people, we can do phase 1 in patients also. So normally phase 1 is done in healthy people, but anti-cancer drugs and all the drugs which are known to be very toxic, the phase 1 is also done in patients. Now after phase 1 trial, we will come to the point that we cannot give a dose higher than this. Now we will go to phase 2, and phase 2 is done in patients. So it is first clinical trial in patients. Now because it is done in patients, so we can know the efficacy. So it is the first trial in which we can know the efficacy of it. So here we will give the drug to small number of patients, usually 20 to 200 patients. And after giving the drug, we will test for efficacy and safety of it, whether drug is effective or the drug is safe. The prime aim of phase 2 clinical trial is to know the efficacy, and side by side we will note the safety also. Now if a drug is found to be safe and efficacious in phase 2 clinical trials, can now we sell the drug? The answer is still no. The reason? We have tested the drug only in 100 to 200 people. It is just giving a hint of efficacy, but we are not 100% sure that it will be effective. So the important thing to remember, phase 2 trials are giving the indication of efficacy, but they are not confirmatory trials. So after phase 2 trial, our answer what we get is the drug may be effective. That we are not 100% sure. So we need to confirm that, and that is done in phase 3 trials. So in phase 3 trials, it is done in same, that means patients. We do the same thing, that means efficacy and safety. But the difference are two. One, there are large number of patients. We do it in thousands of patients, usually 2,000, 3,000, up to 5,000 patients. And second, it is done at multiple centers. We enroll the patients from all over the country, sometimes from different countries also. Now, why to do these two differences? First, why in large number of patients? We already said to confirm the efficacy. Because if the drug is found to be safe and efficacious in 5,000 patients, most likely it will be efficacious. So the difference between phase 2 and phase 3 trials are, phase 2 are indication of efficacy, whereas phase 3 are confirmation of efficacy. Now why to do in multiple centers? To cover the different genetic makeup. So phase 3 trials, they are done in large number of patients taken from different centers to confirm the efficacy of a drug in different genetic makeup patients. So if a drug is found to be safe and effective in phase 3 trials, now we are 100% sure it is safe and efficacious. Now can we sell the drug? The answer is again no. The reason? We require the certificate, we require the license to sell the drug. So what we do is, now we will write an application to the licensing authority, which is called FDA, Food and Drug Administration. Now what is done is, even before the clinical trials starts, we have to write an application to FDA. So there are two type of applications which are written to FDA. One is the application which is written before start of clinical trial, which is called as INDA. It is Investigational New Drug Application. So what we do in this application is, this is before starting the clinical trials, after doing the animal testing. So we have tested the drug in the animals, then we write an application to FDA. Along with that application, we will send all the animal data. So investigational new drug application is written to get the permission to start the clinical trial. That means we are writing to FDA that we have tested in the animals, now we want to test it in humans. So that is called investigational new drug application. Now FDA will give the permission, and then we can do phase 1, 2 and 3 trials. Now after phase 3 trials, we will write another application which is called NDA, New Drug Application. So from the previous application, the main letter which is missing is I. That means investigational. So here the drug is no more investigational. We have already investigated the drug. So now it has become the new drug, and now this application is written to get the permission to market the drug. So we are writing new drug application or NDA to FDA to get the permission to market the drug. So if FDA approve this application, now the drug will come into market. So after the drug come into market, then we will do phase 4 trials. So So phase 4 trials are also called as post-marketing studies because the drug has already come into market. Now what we will do in phase 4 trials? Phase 4 trials, they are mainly done to know the rare side effects of a drug. Rare side effects of a drug. Now we can take very good example. There was a drug called as rofecoxib. This drug was made as an alternative to aspirin. Aspirin, we all know. Aspirin is a very good anti-inflammatory drug, but the main problem with aspirin is it causes GI toxicity like peptic ulcer. So many people who take aspirin on long-term basis, they develop GI toxicity. So to prevent the GI toxicity, we have developed a new drug called rofecoxib. Rofecoxib is aspirin type of drug, means anti-inflammatory drug, but it was not causing peptic ulcer. So when we tested the drug in phase 1, 2 and 3 trials, it confirms that it is effective anti-inflammatory, but it do not cause peptic ulcer. So then NDA was written to FDA and it was approved. So rofecoxib came into market. So whenever rofecoxib came into market, almost all doctors start prescribing rofecoxib instead of aspirin because it is effective and it is safe. Now in 3 years, when the post-marketing studies were done after the marketing of a drug, thousands or in fact millions of people received rofecoxib. The post-marketing studies confirmed that it is not causing peptic ulcer. But the post-marketing studies confirmed that it is causing myocardial infarction. The question is, rofecoxib is causing myocardial infarction. Why we did not come to know in clinical trials, phase 1, phase 2, phase 3 studies? Why it has come only in phase 4 trials? The reason is, myocardial infarction is a rare side effect of rofecoxib. By definition, a rare side effect is that which is seen in less than 1 out of 20,000 people. Means when we give a drug to more than 20,000 people, only one develop that side effect. And rofecoxib, it was causing myocardial infarction in very few people. And as we have seen that maximum 5,000 patients we have tested in phase 3 trial. So maybe that person is missed. But when the drug is given to millions of people, then thousands will develop myocardial infarction. And if we do not do the phase 4 clinical trial, we can never come to know about this. So phase 4 studies are must to know the rare side effects of a drug. And apart from rare side effects, other thing we can do is chronic side effects of a drug. Some drugs cause side effects when they are used for many years. So they can be picked up only after the drug has come into market. So rare and chronic side effects, they are tested in the phase 4 clinical trials. So these are the important points regarding the clinical trials. A quick recap of the important MCQs regarding the clinical trials. Number 1, which is the first phase in humans? It is phase 1. Which is the first phase in patients? It is phase 2. Then in which phase first of all we come to know about efficacy of a drug? It is phase 2. In which phase we confirm the efficacy of a drug? It is phase 3. Which phase contains maximum number of patients? It is phase 4, millions of people. Then in which phase of clinical trial is done after marketing of a drug? It is phase 4 trial. These are some of the important MCQs from the clinical trials.
So we have discussed the basics of clinical trials already. So here we will like to discuss little bit details about the clinical trials. First of all, we'll like to understand some regulatory authorities. There is a term known as licensing authority. Licensing authority means which has the power to give license. In case of America, the name of the authority is USFD, Food and Drug Administration. The similar authority in India is CDSCO, Central Drug Standard Control Organization. Central Drug Standards Control Organization, and it is headed by DCGI, Drug Controller General of India. So these are the licensing authorities. So when we want approval of some drug whole of the world, then we need the permission of FDA. Whereas if we want to use the drug only in India, we need the approval of DCGI. Clear? Now, first of all, whenever we want to start the clinical trial, we want the permission of these people, and we will write an application to these people which is known as investigational new drug application. Investigational new drug application, also known as INDA. So this application we write to the licensing authority. Along with that, we send the animal data. So animal data people will animal data will be studied by these people, and if they think that it can be good for humans, then they will give the permission to start the clinical trial. So INDA is written to start the clinical trial. That means if INDA is approved, we can start phase 1 trials. Okay. So then we do phase 1 trial, phase 2 trials, phase 3 trials, as we discussed earlier. And after the phase 3 trial, when we are sure that the drug is going to be safe and effective in humans, then we will write another application to these people, which is known as new drug application, NDA. Now, how this application is different from earlier? Earlier was IND, here it is NDA. So basically here I word is not there. That means the drug is no more investigational. It has already been investigated into. So now it has become the new drug. So NDA is written to get the permission to market the drug. So in this, we ask to market the drug. We send the data phase 1, 2, 3 trial. They study the data, and if they think that it is good for humans, then they will give the permission to market the drug. So that means after the approval of NDA, the drug will come into the market. Okay? So two implications. One is INDA, second is new drug application. So one is this. Now, second important thing to remember about the ethical guidelines. So we need to study the phase 1, phase 2, phase 3 and even animal studies. They should be done in such a way that the data which is generated, it is credible, and also they have not caused any harm to humans or animals. So which authorities see this? So for animal studies, preclinical studies, also called as animal studies, the controlling authority is CPCSEA. So it is committee for the purpose of control and supervision of experiments on animals. So CPCSEA is committee for the purpose of control and supervision of experiments on animals. So before doing any animal study, we should get the permission of CPCSEA, okay? Committee for the purpose of control and supervision of experiments on animals. Okay? So after getting their permission, we can do animal studies. Basically, the purpose of these will be that the animals are not harmed unrequired. Clear? So basically ethical issues are handled. Whereas for clinical trials for human studies, there is guidelines known as GCP guidelines, Good Clinical Practice Guidelines. Good Clinical Practice Guidelines. So there is a booklet of GCP guidelines which tells every step how to go about ethically. So if the studies are done according to GCP guidelines, they are good quality studies. They have high chances of getting approved because the data are generated is credible. Yeah. So these are the authorities which see the ethical issues. Okay. Now some little bit of detail about these phases. Phase 1 studies we already discussed. It is done in healthy people, and sometime if the drug is known to be very toxic, we can do in patients also. The major aim of phase 1 trials we already discussed is to know the maximum tolerable dose, MTD, maximum tolerable dose. Okay. Now phase 2 studies, these are done in patients. First of all, we know the efficacy. Now, special thing to remember, phase 2 studies may be phase 2A, they may be phase 2B. Phase 2A studies are known as proof of concept studies. Proof of concept study means we want to first of all know whether the drug will work or will not work. So first of all, we wanting to know that whatever concept we have come with, we are proving that concept. So that's why they are known as proof of concept studies. So first of all, we will see whether the drug works or not. Suppose the drug starts working. 100-200 people we have taken, the drug is safe and efficacious in these. Then in phase 2B, we can do dose ranging studies. Dose ranging studies, that means we can take different doses, but all the doses will be below the maximum tolerable dose and see the effect of different doses. That is called dose ranging studies. Okay? So phase 2. Then phase 3, which is done in large number of patients, thousands of patients. The other name of that is pivotal trials. Phase 3 are called pivotal clinical trials. So basically they play the pivotal role for the approval of a drug. So that means they are the confirmatory. Pivotal means simply they are the confirmatory studies. So these are the different names of phase 1, 2 and 3. And phase 4 we all know, they are after the marketing of a drug. So they are called as post-marketing studies. Post-marketing studies. Now in the clinical trials, there is a separate study which is called as phase 0 study. Phase 0 study. The other name of this is micro dosing study. Now, important thing to remember, many drugs, they fail even after development. So we have started developing a drug, we have already spent several $1 million on that drug, and that drug fails. So drug development is very costly affair. Out of 10-20 drugs when we develop, only one can be approved, most of them fail. So whatever money we have spent on those drugs which have failed, that all is a waste. So pharmaceutical companies thought that if we can come to know why a drug fails, so we will not develop that drug which has high chance of getting failed, so we can save on our money. So they tried to find out why a drug fails. So they came to know the two major reasons. The first reason is the drug is not getting concentrated at the place where we want. Like we want the action of a drug in liver, but the drug is not going enough in the liver. So the drug may not be effective. And second reason the drug fails is the drug is getting concentrated at some area where we do not want. Like we want the action of the drug in the liver, but it is concentrating in the retina. So it can cause adverse effects there. Okay? So pharmaceutical companies thought that if we come to know beforehand whether a drug is going at which places, then we will develop only those drugs which have favorable pharmacokinetics. Okay? So then they came out with a plan to carry out such studies which are called phase 0 studies. So what is done in this, as the name tells, the drug will be given in sub-therapeutic doses, micro doses. Okay? So what we do is we give maximum of 100 microgram of the drug, or we give 1/100th of the dose which we want to use clinically, human equivalent dose. So 1/100th of the human equivalent dose, or 100 microgram, whichever is lesser. So out of these two we will give. Now important question, when we are giving so small dose, will you expect that the drug will be efficacious? Obviously not. So we cannot know efficacy in phase 0. Can the drug produce side effect at this low dose? Again, the chances are negligible because we are giving sub-therapeutic doses. So we cannot know the safety or toxicity of a drug also. So we cannot know efficacy, we cannot know safety. So what we will do then? So what we do is in phase 0 studies, we will take very small amount of a drug and add a radioligand. So we will add a radio labeled substance to this. And then we will sit and see the counter wherever radioactivity is going. So we can come to know the drug is going to which places, where all it is concentrated. So only those drugs will be developed which has favorable pharmacokinetics. Okay? So phase 0 studies, it is done in humans. First of all, it is a clinical trial, so it is done in humans. Another name is microdosing study. We cannot know the safety, we cannot know the efficacy, but we can know the pharmacokinetics, where the drug is going. And the maximum dose is 100 microgram. Usually we give 1/100th of the human equivalent dose. And most importantly, these studies are not mandatory. Pharmaceutical companies are doing themselves to save their own money. It is not mandatory. Okay? So that is about phase 0 clinical trials. Clear? One more thing we can discuss here is control and blinding. Suppose we want to develop a new analgesic drug. So we have developed a new analgesic drug, painkiller drug. We give to 10 people, and out of the 10 people, 5 people say that the pain is less, and 5 people say that pain is not affected much. Is the drug efficacious? We cannot say. Suppose out of 10 people, 8 people say that the pain is no more present, and 2 people say that still pain is there. Is the drug efficacious? Yes? No. Still we cannot say. The reason is, you will see many times when you go into the wards, so you have a night duty, you are intern, you are alone in the ward. A patient's relative came to you at 2 a.m. saying that डॉक्टर साहब बहुत ज़्यादा दर्द हो रहा है हमारे पेशेंट को, वेरी सीवियर पेन इज देयर, एंड यू आर इंटर्न, you do not know much, you do not know what happens if you give some drug. So what you will do, many of the times what we do is, we ask the sister, give normal saline to this person. So when sister give the normal saline, 8 or 9 patients out of 10 says that now pain is not there, they sleep. So this effect is known as placebo effect. So that means the 8 people which we are getting the response, they may be getting the placebo effect. So just the feeling that the person has been given some substance, some drug is given, so just that feeling may terminate the pain, which is known as placebo effect. Placebo means I shall please. So placebo effect may be there. So we want to know whether this is the actual effect of a drug, it is a placebo effect. So to know that, we need to keep a control group. So whenever we do the study, we will keep the 2 groups, one drug group and second control group. Control group, mostly we keep it as placebo, but sometimes using a placebo is not ethical. Like a patient, we are wanting to try a new drug for myocardial infarction. So what we do, we take 20 people, to 10 people we give the new drug which we have developed, and to 10 people we do not give any drug. Those people will die. So we cannot keep placebo here. So when we cannot keep the placebo, so here we will give active standard, or we can say standard control. Standard control means the already known existing treatment for that condition. So placebo basically, how to make placebo is, it is a drug which has everything similar to drug. That means the color is same, size is same, smell is same, everything is same. The only difference is it do not contain the active ingredient. So it can also be called as dummy medication. Dummy medication, placebo is a dummy medication. So it looks like the drug, but it do not contain the active ingredient. Clear? So placebo, it is made, or standard drug control is taken, and in 2 groups we are doing. Sometimes placebo can be in the form of surgery also, like we want to compare the surgery with the vis-a-vis without the surgery. So again what we will do, we do sham surgery. That means we open the abdomen and do not do that surgery and close the abdomen. Clear? So that is placebo, sham surgery. Clear? So placebo is there, and placebos do not produce effect in everyone. We do not know in which person it will produce the effect, in which person it will not produce the effect, and it is assumed that the effect of placebo, mostly they are due to release of endorphins in the body. Endorphins are released, they give the good feel. Clear? So placebo do not produce effect in all the patients. In some they may produce, in some they may not produce the effect. So what we do is, we have given taken 20 patients. To 10 patients we give the drug, new drug, and to 10 patients we give a drug similar, everything similar to the drug except that it do not contain the active ingredient, that means placebo. Now what is the important thing is, we have given to 10 people drug, to 10 people placebo, and in every person we tell that you are getting the drug and you are getting the placebo. So our purpose will be lost, because if a person come to know that I am getting a sugar tablet, I am not getting a drug, so obviously the placebo effect will not come. So important thing is, these people should not know that who is getting a drug, who is getting a placebo. So everyone should assume that I am getting a drug. So this is known as blinding. So blinding is very important. We keep the person blind. Blind means that person is unaware of the treatment. What I am getting, the person do not know. Remember, in clinical trials, just like in normal language, we use the word doctor and patient. In clinical trial, the word is not doctor and patient. In clinical trial, the name doctor is replaced by investigator, whereas the patient name is subject. So both subject and investigator. Subject means who is receiving the drug, and investigator who is analyzing that, investigating that. So there are two type of blinding. One is known as single blind studies and second is known as double blind studies. So in single blind studies, the subject is unaware of the treatment. So subjects do not know which is getting drug, which is getting placebo. That is called as single blind studies. And single blind studies are usually done in phase 2. Phase 2. On the other hand, in double blind studies, both subject and investigator are blind. They are not aware of. Now subject we can understand, because if he come to know that I am getting a sugar coated tablet, then the person will not develop placebo effect. So logical, we should not tell the subject. Now why we do not tell to investigator which person is getting a drug, which is not? So yes, to remove the investigator bias, because in my mind there may be bias that this person is not getting a drug, so he should not get the effect. So bias in the result may occur. So that is called investigator bias. So double blind studies will eliminate the investigator bias, and these are considered to be the best studies. So they are done when we want to confirm the results. So double blind studies are done in phase 3. Phase 2 are single blind studies, phase 3 are double blind studies. And what is about phase 1? Phase 1 are not blinded. We don't need to blind, because here we are not seeing the effect. We are just checking the toxicity. We are not giving a drug and seeing the efficacy of a drug. So phase 1 studies are known as open label studies. Open label means neither subject nor investigator is blind. Clear? So these are the special points about the high 5 points about the clinical trials. Clear?